Hello, and welcome to this video tutorial, in which I will describe how to use a tool to very quickly generate a batch of DNA parts from a set of protein sequences which you want to incorporate in an iGEM project. Here we have a file with 3 protein sequences. They are all in FASTA format. Some information on the gene or the protein, and the actual amino acid sequence of that protein. We will load up the Protein to Biobrick design tool. Here we get to select the desired assembly standard. This will influence which restriction enzyme sites the program will make sure are not present in the body of the sequence, the DNA sequence for the part. We also have an option to add the appropriate prefix and suffix for that assembly standard. A job ID, number, or label can be given. This will be used to name the folder containing all of the files generated by this program. And finally, a button here enables us to open the FASTA file containing the protein sequences. Here I'm going to look through these folders to find the file I featured earlier in this video containing the FASTA formatted protein sequences. I will open this and the program will do the rest. In a flash, we have a new folder which contains 3 files. First, a report on what the program actually did. Looking through sequence 1, it checked first for the EcoR1 restriction site, the XBA1, the SPE1, the PST1, and it found actually a not1 site. It modified this sequence and then checked again. So on the second pass, it did not find the not1 site because the single not1 site that was present had been changed. This was done for the first, second, and third sequence in the file. Some information about the run is contained here. For example, which assembly standard was used, whether or not the prefix and suffix was added, and the total amount of runtime it took this program to carry out these steps. The results are shown here. The prefix and the suffix are shown in uppercase letters. The resulting DNA sequence is shown in lowercase letters. And this has been done for all 3 genes. As a quick way to verify that this altered DNA sequence, or this DNA sequence, which does not contain the assembly standard restriction recognition sites, to make sure that it actually does encode for the protein, that sequence has been already back translated and is available here for comparison with the original protein sequence.